Ancient bacteria could be sleeping beneath the surface of Mars, where they have been protected from the harsh radiation of space for millions of years, new research finds. While no evidence of life was found on the red planet, researchers simulated conditions on Mars in a laboratory to see how bacteria and fungi might survive. Scientists were surprised to discover that bacteria could probably survive for 280 million years if buried and protected from the ionizing radiation and solar particles bombarding the Martian surface. The results suggested that if life ever existed on Mars, the dormant evidence for it may still be found beneath the planet's surface, a location that future missions could explore when they drill into the Martian soil. While billions of years ago Mars was likely a more hospitable environment for life, complete with an atmosphere and water on its surface, today the red planet looks more like a frozen desert. The planet's arid mid-latitudes have an average temperature of -80 degrees Fahrenheit, -62 degrees Celsius. And then there's the constant danger of radiation because Mars has such a thin atmosphere. There is no flowing water or significant water in the Martian atmosphere, so cells and spores would dry out. Study co-author Brian Hoffman, Charles E. and Emma H. Morrison, professors of chemistry and professor of molecular biosciences at Northwestern University's Weinberg College of Arts and Sciences, said in a statement. It also is known that the surface temperature on Mars is roughly similar to dry ice, so it is indeed deeply frozen. A research team determined the limits of survival of microbial life when exposed to ionizing radiation, as might be the case on Mars. Then the team introduced 6 species of bacteria and fungi found on Earth into a simulated environment on the surface of Mars, and simultaneously irradiated them with protons or gamma rays to mimic space radiation. Deinococcus radiodurans emerged as the clear winner. Nicknamed Conan the bacterium, for its tough nature, the microbe seemed perfectly suited to life on Mars. The bacterium is polyextremophilic, meaning it can survive harsh conditions such as dehydration, acidity, and cold. The hardy microbe is one of the most radiation-resistant organisms known to science. Previous research has found that the bacteria could survive 1.2 million years just below the surface of Mars amid the harsh radiation and dry, frozen environment, and some microorganisms known to survive millions of years on Earth. The new study found that if the Conan bacterium were dried, frozen and buried deep beneath the surface of Mars, it could survive 140,000 units of radiation, 28,000 times the level of radiation that could kill a human. The bacterium, which resembles a pumpkin under a microscope, would likely survive on the surface of Mars for only a few hours after being exposed to relentless ultraviolet light. The expected survival of the bacterium Conan increased to 1.5 million years just 10 centimeters below the surface and to about 280 million years when the bacterium was 10 meters deep. The researchers were able to measure how many manganese antioxidants accumulated in the cells of the microorganisms when they were exposed to the radiation. The more manganese antioxidants the team found, the more likely it was that the microbe could withstand the radiation and survive. The genomic structure of the Conan bacterium links chromosomes and plasmids together, meaning cells can stay aligned and repair themselves after exposure to radiation. And if a microbe similar to Conan evolved on Mars billions of years ago, when there was still water on the surface of Mars, the slumbering remnants of the bacteria may just lie dormant deep in the planet's subsurface. Although D. radiodurans buried in the Martian subsurface could not survive dormant for the estimated 2 to 2.5 billion years since flowing water disappeared on Mars, such Martian environments are regularly altered and melted by meteorite impacts. Study author Michael Daly, a professor of pathology at the Uniformed Services University of the Health Sciences and a member of the National Academy's Committee on Planetary Protection, said in a statement, We suggest that periodic melting could allow intermittent repopulation and dispersal. Also, if Martian life ever existed, even if viable lifeforms are not now present on Mars, their macromolecules and viruses would survive much, much longer. That strengthens the probability that, if life ever evolved on Mars, this will be revealed in future missions. The results have implications for both returning Martian samples to Earth and landing manned missions on Mars. The Mars Sample Return Program, an ambitious program led jointly by NASA and the European Space Agency, will launch multiple missions to Mars to collect and return samples collected by the Perseverance rover. The rover team hopes rock and soil samples taken at the site of an ancient lake and river delta in Mars' Jezero crater could determine if life ever existed on the red planet. The samples could even contain microfossils of ancient microbial life. Additionally, astronauts have the potential to accidentally hitchhike bacteria from Earth when they land on Mars. We concluded that terrestrial contamination on Mars would essentially be permanent, over timeframes of thousands of years, Hoffman said. This could complicate scientific efforts to look for Martian life. Likewise, if microbes evolved on Mars, they could be capable of surviving until present day. That means returning Mars samples could contaminate Earth.